In the last lesson, we learned exactly how an individual gene gets turned into a protein. This genetic code we analogize to a book of recipes representing our genome. We want to revisit this analogy for this lesson, but we're going to think about it a little bit differently. Instead of these ingredients being used to make a single protein, we're going to take each of these ingredients individually as its own protein to build a particular trait or even a whole human being. Let's get started. And if we want to build a human being, the first thing I'm going to do is take our ingredient, which represents one protein, and I'll start measuring out one tablespoon.
Okay, so this is one protein?
That's right.
Well, one human coming right up.
Lauren, did you just pour all of the flour in the bowl?
I mean, yeah, you said flour was in the recipe, and I'm assuming it takes a lot of flour to make a human. We're huge.
This is true, but like a gene, you can't just add the whole thing at once. You got to add the right amount at the right time to get the right product.
Well, I'm sorry, but I guess you're right.
There are over 20,000 genes in the human genome. If we added all of them all at once, all at the same time, every cell would be identical because they'd all have the same ingredients at the same time. And obviously, that's not what we want. To get different cell types at different times, we actually need to add different ingredients at different times and in different amounts. So we must specify these ingredients, and we have to specify the amounts.
Well, where does the specificity come from? Like, how does a skin cell know to be a skin cell, and how does a brain cell know to be a brain cell?
The short answer is development. Development is a process by which the right gene is added at the right time to make the right cell type. It's how one cell knows to be a certain cell type out of the many thousands it could possibly be. For example, the cells in your hand.